Okay, so we talked about how you can make an antibody really specific to an antigen using VDJ genetic recombination. And then in the previous video, we talked about how you can kind of change the codes of an antibody, essentially isotype switching, so splicing out different constant regions to give different shapes of antibodies. One thing I kind of mentioned but didn't really talk about in much detail is that you can change the site of the antigen binding region even more by introducing mutations in the VDJ segments of the genes to make it even more specific, kind of by chance, to antigens. This is the thing, remember, that drives avidity to affinity. What was avidity again? So avidity is like your fishing net. It's not as specific to your fish, to your antigen, but your overall binding strength is kind of much stronger. Whereas affinity is like taking the time to study a fish and like study the bait and kind of perfect it. And therefore, the bond between the fish and the fishing rod is much stronger, and therefore you don't need as many fishing rods. So that's kind of why you have loads of IgM, whereas not so many IgG. The process here of somatic cell hypermutation is the thing that makes this fishing rod even more specific. Basically, it's the mechanism that makes these mutations in the VDJ regions so that your antibody and your antigen bind really, really tightly. Okay, so imagine we have our like a skin and our epithelial cells here and our blood here, and unfortunately we're going to get an infection. So pathogens is going to come here, all with the antigens in like the epithelial kind of tissue or your non-lymphoid tissue. And also you've got your blood, and say some of the antigens make their way into the blood, but hey ho, look, there's a B cell that's kind of just patrolling, and through VDJ recombination it has a B cell receptor, so an antibody that just so happens to fit in with this antigen, and so these guys are now bound. Interesting. So what's going to happen is this B cell is going to migrate to lymphoid tissue, which is specialized immune tissue. This immune tissue has a bit which is called the germinal center in it, and the germinal center has a dark zone and a light zone. So B cell has migrated into here, and what it does is it goes to the dark zone first of all, and it proliferates like crazy. So basically this is your kind of like monoclonal activation. Your B cells can't proliferate in the blood or next to other normal cells, because what happens is your normal cells that the B cells are next to kind of get jealous, like, why is the B cell proliferating so much? And all those kind of hormones that make your B cells proliferate, if they touch other cells, your other normal cells will start proliferating like crazy and you get stuff like cancers. So that's why your B cell has to go into seclusion, into the germinal center in the lymphoid tissue and proliferate there. Don't worry about the numbers for now, but there's something that I missed out, which we'll go through afterwards. So in the dark zone, the B cells are proliferating, but that's not the end of the story. There's also stuff that makes them mutate. There's kind of chemicals that makes them mutate like crazy, and as they're proliferating, they're also going to mutate. And obviously, each genetic recombination, each genetic mutation is going to alter the antibody shape essentially, and we're going to end up with different antibody shapes each time. So this causes obviously different FAP regions. Okay, so meanwhile these guys are proliferating and mutating simultaneously. So this is going on, but in the back of your mind, we've also got the antigens here still, and essentially there's a type of cell called a dendritic cell, which is kind of like an octopus. It has all these long kind of tentacles, and this is in your infected tissue. And what it does is it grabs, using its tentacles, it grabs onto these antigens, and is able to migrate into the immune tissue, which is useful later on. So it migrates into the light zone, basically it grabs the antigens and goes all the way up via the blood, via the lymphatic system, into the light zone, and it's also going to go in also in your lymphoid tissue in general, but we'll talk about that later. Okay, meanwhile, we're still here. These B cells are still proliferating like crazy and still mutating. What's going to happen is, before anything further happens, some of these B cells, now they've proliferated, we've kind of got our monoclonal activation, monoclonal expansion. So some of those are going to now leave your dark zone and then become memory B cells or plasma cells. However, some of these B cells, the proliferated ones and the mutated ones, will actually go onto the light zone of your germinal center, this is all like specialized lymphoid immune tissue. And in the light zone, look who it is. It's the dendritic cell, which has come, like here's one I made earlier, and just so happens to be waiting for them with all these antigens. It's actually quite cool. So what happens is that the dendritic cell has all these antigens, and all these B cells have mutated to have different receptors. Hopefully the aim is that some of these receptors will fit onto the antigen even more. So the dendritic cell is like, yo, it's time to test it out. Let's see who can bind to the antigen strongest. So essentially, strongest man wins, and whoever binds to the strongest will go into the dark zone again, proliferate and mutate again, and perhaps it will introduce a mutation that makes it bind even more stronger. The ones that don't bind strongly, they'll just get killed off by apoptosis, so you get rid of that kind of cell line. And then therefore you get, essentially you're mutating and mutating, you're evolving this antibody to get super, super strong connections until hopefully we get an antibody, which is really, really nice, like our fishing rod, and fits really nicely into our antigen. Okay, So one of the functions of the dendritic cells then is to bring antigens into the light zone to kind of test out the different antibodies. The other function, so your dendritic cell is also going to come into your, this all here is your lymphoid tissue. So it's going to come into your lymphoid tissue, and in your lymphoid tissue is your T helper cells. And your T helper cells, they're essentially the cool kids. They're too cool to be hanging out in your infected tissue. They just hang out in your immune tissue with their cool friends. So in order for them to know that an antigen is here, they need the dendritic cell to come out, grab the antigen, and transport the antigen to them. They're kind of lazy, so they don't go to the antigen has to come to them. But when the antigen comes to them via the dendritic cells, so this is like another dendritic cell, it's not the same one as this. Another dendritic cell will present an antigen to a T helper cell, the cool kids, and then it's like, okay, I need to stop being lazy. It wakes up, and it kind of caterpillars into butterflies into a follicular helper cell. So by the way, this germinal center is called a follicular center as well, so that's why it's called a follicular helper cell. And this follicular helper cell is able to go into the light zone, so it's made in the, it turns from the helper cell into the follicular cell in the lymphoid tissue, and the lymphoid tissue, it goes from the lymphoid tissue, it goes into the light zone of the germinal center. And what it does there is essentially it monitors this kind of process of seeing which B cell is the most attracted to the antigen. Essentially, the T follicular helper cell acts kind of like as a referee for step 6. And essentially, so this happens over and over again. The B cell, under the refereeing of a follicular helper cell, the B cell that binds most tightly to the dendritic, to the antigens on the dendritic cell, will go back into the dark zone. The other ones will die, mutate even more, and then go back here. And this can happen loads and loads of times until we get a fit that's really, really nice. So essentially, we've introduced these mutations that have changed the shape of the antibody receptor. And just by chance, we're getting stronger and stronger binding, and that process is repeated. Okay, just to brief, and this process is called affinity maturation. We're getting closer, more and more mature to more affinity, and so less like our net and more like our affinity. And that's why after, a few days or like a week or two, then we're able to have a really nice fishing rod. We don't no longer need our IgM, which is less specific, because now we've got a really nice specific antibody by introducing mutations, random mutations to kind of evolve our antibodies, and therefore we can have a more an antibody with high affinity. Okay, very briefly to talk about how exactly does this T follicular helper cell referee this. So we knew that the way it becomes a follicular helper cell in the first place is that a T helper cell gets antigens presented to it by another dendritic cell, so not this one, but another one. And this T helper cell wakes up and caterpillars the butterflies into a beautiful follicular helper cell. This follicular helper cell is able to go into the light zone and is essential for the affinity maturation of antibodies. So all of that is stuff that I've said before. Okay, and what they do is they're going to secrete a hormone, an interleukin, called interleukin 21. And that is essentially responsible for the B cells to proliferate and also to differentiate in the germinal center. Another thing as well is that once your B cell has bound to your antibody, once your B cell has bound to your antigen, the one that has bound tightly, there's always two-step verification. So one method of two-step verification is your B cell that has bound to your antigen. See, I get so confused with this. Your B cell that's bound to your antigen very tightly is going to express a kind of lock called a CD40. And this receptor is another additional receptor on its surface. And your follicular helper cell has the key to that lock, which is the ligand of CD40. It clicks in, fits in, and then your two-step verification is complete. And this allows your this essentially allows your B cell to have that affinity maturation. So in a nutshell, the follicular helper cell is really, really important for your B cells to proliferate in your dark zone and also for this affinity maturation. Also, T follicular helper cells also drive the isotype switching, so they also produce lots of interleukins that are responsible for determining whether this B cell, when it becomes a plasma cell, is that going to be an IgA, an IgE, etc., an IgM, etc., IgG. And finally, we have some questions.